Science friends. We are going to do a little lesson today on DNA, RNA, and then how proteins are made. So a lot of this we talked about in Unit 1 when we learned about DNA there. But I'm just going to go through all of it again. But bear in mind, this will be a bit of a longer lesson. I would highly encourage that you take notes on your own paper. There is a 4-minute video on my PLTW that explains all of this, but it does so very, very rapidly and quickly. And so hopefully you can get a little bit more of it as I kind of walk through it a little bit slower. The video is excellent and it's a good short review. But if you're wanting to get a little bit more and try to understand it better, then you're going to get more from this video. And then you'll have a short little assessment in Google Classroom today. So you'll also want to make sure you read the intro, and I think it's step 1 or step 2 that has some of the functions of proteins, and take some notes on that because that's not something I'm going to really touch on today. But that's important for what we're doing right now in our lesson, is understanding what proteins are and what are some of their functions in the human body. But today we're going to focus on DNA and RNA and then how proteins are made. So we talked about before, DNA are made up of units, is made up of units called nucleotides. And these nucleotides have 3 parts. So you've got your 5-carbon sugar. That's drawn here. Each of these, it's a pentagon because when you draw sugar, the points represent the carbon. And so if a pentagon has 5 sides or 5 points, and so those each represent a carbon. In deoxyribose, that sugar is, or in DNA, that sugar is deoxyribose. In RNA, that sugar is ribose. So you've got your 5-carbon sugar, your phosphate group, and your nitrogen base, okay? Your nitrogen base. And so those nitrogen bases are A, T, G, and C, and we'll talk about those more in just a minute. But you make a nucleotide, and then nucleotides repeat, or they build together to make up a nucleic acid, such as DNA or RNA. So going on to the bases, there's 4 types of nitrogen bases, as I just mentioned: A, T, G, and C, adenine, thymine, guanine, and cytosine. And so the adenine and guanine are purines. That means they have a 2-ring structure, versus the cytosine and thymine are pyrimidines. They have a single ring structure. Also uracil, if you want to write that here, that is a special nitrogen base that's only found in RNA. And we'll talk about that a little more again later. But you see, so this is one nucleotide. Sugar, phosphate, base, okay? Another nucleotide, phosphate, sugar, base, all right? And so when these pair up, you have the bases that pair in the middle. A pairs with T and C pairs with G, and then you have the sugar and the phosphate that alternate in the backbone, okay? The bases are held together by a hydrogen bond, which is a relatively weak bond because when the DNA sometimes needs to be broken apart down the middle, and so those bonds need to be able to be broken easily. But these bonds are a different type of bond called a covalent bond, and those are much stronger. We don't want to break those bonds apart quite as easily. But today we're going to talk about some of the instances when these hydrogen bonds get broken apart. One thing that you may also notice about this is that here the 5-carbon sugar, the point goes up, right? But over here, the point goes down. And so this is referred to as the anti-parallel nature of the DNA molecule, how one strand seems to run in the opposite direction of the other strand, okay? And these bases, though they have to be paired A and T and G and C, they can be paired in any order, okay? So this one shows A, G, T, A, C, G, but you can make that any order. And that's going to be what codes for different proteins, the order of the nitrogen bases. Okay? So, as we mentioned just briefly, we have Chargaff's rule that tells us that A pairs with T and G pairs with C. So this guy down here is the name of Erwin Chargaff, and he was the one that discovered this by looking at samples of DNA and realizing that the percentage of guanine was the same as the percentage of cytosine, and the amount of adenine was the same as the amount of thymine, and that's because those always pair together. Okay, so going back to this previous picture, adenine pairs with thymine, and guanine pairs with cytosine. So you see you always have a purine paired with a pyrimidine, a 2 ring structure paired with a 1 ring structure. And that makes sure that these bases in the middle are always the same length apart. Okay, you don't see the rings shown here, but if you've got a 2 ring paired with a 3 ring, then it's always going to be 5 rings essentially apart. It doesn't work the other way, because then your DNA would go like in and out and in and out, and your ladder would be not right. Okay, so this is just another picture of our structure of DNA. You've got a nucleotide with the sugar, phosphate, and the base. Your base is in the middle that hydrogen pair together according to Chargaff's rules, and then your sugar and phosphate that make up that backbone. Okay, notice here you see the G and the C pairing together, and they have a different fit. The A and the T wouldn't fit with the G or the C, so they fit together specific. Okay, so in a eukaryotic cell, which is a cell that has a nucleus, such as all human cells are eukaryotic cells, DNA resides in that nucleus, and we see it in the form of chromosomes. So we've talked about chromosomes last week. So this is a chromosome here, but the DNA is coiled up around these proteins called histones. It helps to wind that DNA up and wind it in these coils, which then coil into a tighter coil called a supercoil, and it packs all of that DNA, that genetic material, into the chromosome. Okay, but remember, you only see the chromosome during like mitosis, when the cell is dividing. That's when the chromosomes condense. The rest of the time, the DNA exists just as the chromatin or the uncondensed form. But a DNA molecule is very long. In the human DNA, you've got over 1 meter of DNA. Actually, we got closer to 2 meters. It's about 6 feet of DNA is how much DNA is packed in every single one of your cells. Okay? So this just explains that chromatin structure and the histones, as I just mentioned a moment ago. Okay, again, take whatever notes that you feel like you need to take. You don't have to write everything down. So, when sometimes the DNA has to copy itself. We have seen that like in mitosis. The DNA copies itself before the cell divides. And that's what he says here. Before a cell divides, it copies the DNA. And that process is known as DNA replication. And so that makes sure that each daughter cell is going to have a whole new set of DNA. And so when it divides, basically what we have is, I'm just going to draw it like a ladder, okay? Our DNA molecule, that's what a ladder is. It looks like a ladder. Normally it's twisted to kind of be a spiral ladder instead of just a straight ladder. But we have both of these strands of the DNA, and they're both referred to as our parent strands of DNA, okay? So our parent strands of DNA are going to separate from one another, okay? Those hydrogen bonds are going to be broken down the middle. Remember, I said those are weak bonds, and we want to be able to break them apart. And then each of these parent strands can serve as the template for a new strand of DNA, okay? And it's made according to Chargaff's rule. So if this one was A, then that one that used to be across from it would have been a T. So that one there would be a T, and this one would be an A, and that one would be a T. Okay, so it's still making the same DNA, but it's going to copy that sequence according to Chargaff's rules. So these new ones are called the daughter strands right here. Both of those two in the middle. I think I put an extra U in daughter there. But those are the daughter strands, and these are the original parent strands. But what you're going to end up with, assuming there's no mutations, which we'll talk about tomorrow, you're going to end up with two identical DNA strands, okay, that are the same as the parent. And so both of the parent DNA splits and serves as a template, is what we call it, for the new strand. And you end up with two new complete sets of the DNA. And that's what we see here, okay? You see the DNA splitting, so this was the original. You're going to try to wind your eyeballs and follow it, okay? Here's the original, the blue part. And you see this orange part here being synthesized, the new DNA, being synthesized according to that base pair rule. So this green thing here is a special enzyme, right there also, known as DNA polymerase. And what DNA polymerase does is he brings in the new nucleotides. So if this is an A, he's going to bring in a T. If that's a G, he's going to bring in a C. Okay? Another A, he brings another T. And so on and so forth to build the new strand. And so this happens in both directions. You know, it's happening on top and it's happening on bottom. And ultimately, we're going to get two new strands of DNA. Okay? This area where the DNA opens up is referred to as the replication fork. And it continues until the DNA is fully copied. Okay? So there's that enzyme we just mentioned called DNA polymerase. What's important to know is pretty much anything that ends in an ACE is going to be an enzyme. We use lots of enzymes in biomed and in biology. Enzymes are proteins. Enzymes are things that speed up a chemical reaction, but they almost always end in ACE. Lipase is an enzyme that breaks down lipids. Protease is an enzyme that breaks down proteins. They frequently break apart other bigger molecules. Okay? Galactase. Lactase is an enzyme that breaks down lactose. If you're lactose intolerant, you don't have lactase. And so your body can't break down lactose and it makes you sick. Okay? So DNA polymerase, though, is responsible for bringing in the new nucleotides, like we see it doing right here. DNA polymerase is also important because it's a proofreader. And so it's making sure that the bases are brought in correctly. And if there's an error, it helps to fix that. Okay? That's the job of DNA polymerase. So when we talk about DNA, we frequently talk about genes. And I talked about those last week, just a little bit. So our DNA molecule, right? I think I talked about those. Maybe I'm getting confused with biology. This is our DNA molecule, but it's got all these letters on it, right? T's and A's and G's and C's and so on and so forth. And it's going to code for thousands of different proteins. The whole DNA molecule does. But one spot on the DNA molecule, which codes for one specific protein, is known as a gene. It's a series or a segment of DNA that contains the instructions to make one protein. Okay? And so when we need to make that protein, we go to that specific spot. Right? It's like a cookbook. You have a cookbook that has, 200 recipes in it. But every time you want to get a recipe, you don't copy the whole cookbook. You go to that one page and you write down that one recipe and you give it to your friend. And so now your friend can make strawberry pie or whatever she's trying to make. Okay, so if we want to make this one protein, we go to our cookbook, that is the DNA that lives in the nucleus, and we copy this one gene that has the instruction for making that one protein. And we copy it down and we send it to the RNA. And then the RNA helps to make it. Okay? And actually, when we copy it down as RNA, and then the RNA makes it into a protein. Okay? So, here's what happens. Here's what you got to know. Tattoo this on your forearm. Okay? DNA to RNA to proteins. This is the big picture. If I had $1 for every time I have written this on my board, I would be a very rich woman, okay? DNA to RNA to proteins. This first step is called transcription. Transcription is the conversion of DNA to RNA. RNA to proteins is referred to as translation. Okay? Translation happens on the ribosome. That's a little organelle inside the cell. Okay? Transcription happens, you can't read this what I'm writing here, but in the nucleus, because that's where the DNA lives. The DNA does not leave the nucleus. So if you want to do anything with the DNA, you've got to go to the vault of the nucleus to mess with it. Okay? So, we know that DNA and RNA are both nucleic acids, deoxyribonucleic acid, ribonucleic acid. We know, as we've mentioned before, that they have different sugars. But there's some other differences between DNA and RNA. There's 3 differences between DNA and RNA. So 1, the sugar in RNA is ribose. We've got deoxyribose in DNA. The second one, RNA is generally single-stranded. Okay? So while we think of DNA as this double helix that you see here in my picture, it's got 2 strands. RNA has only got half of it. So you're not going to have the other side. You're just going to have, one side of your ladder. It's not going to have the opposite of the base pairs. Okay? So that's important with RNA. RNA is single-stranded. And then the last one, which I mentioned at the beginning, is that RNA contains uracil instead of thymine. I talked about that up here just very briefly. Uracil, which is in RNA. Okay? So, we know that normally A pairs with T. T is going to pair with A. G is going to pair with C. And C is going to pair with G. Okay? But in RNA, there is no T. And so A would pair with U. Okay? And you could also switch this one. T would pair with U. Okay? I'm going to show you what I mean in just a moment. Get back here where I'm trying to go. Okay? So, the main job of RNA is to assemble amino acids into proteins. It takes the directions from the DNA, okay, the nucleotide sequence in the DNA has to be copied into RNA, and then RNA uses those instructions to make proteins, okay? DNA to RNA to proteins, transcription and translation, all right? So, when we talk about RNA, there's actually like hundreds of types of RNA, but we're only going to talk about 3 types of RNA. They're all really important. The first is the messenger RNA, and that's probably the most important one that we're going to talk about. When we're talking about this RNA here, that is the mRNA that we're talking about, the messenger RNA. It takes the message from the DNA and uses it to make the protein. Okay? So it's the messenger from the DNA to the rest of the cell, and it has the instructions for making proteins. This should say from amino acids, not into amino acids from amino acids. Okay. The next type of RNA, though, that we're going to talk about is rRNA, and that stands for ribosomal RNA. So remember I said that this step happened on the ribosome? The ribosomal RNA makes up the ribosome, okay? And so it also is important in making the proteins because proteins are made on the ribosome. Translation happens on the protein, or translation happens on the ribosome, and in translation we make proteins. So proteins are made on the ribosomes. That's important. Proteins are made on the ribosomes, okay? Now, the last type of RNA that we're going to talk about is tRNA, which is transfer RNA. And so it is important for transferring amino acids to the ribosome, okay? So, if you think about a protein as a strand of pearls, all right? But let's think of our pearls are like multicolored. They're not all the same. It's not a beautiful white pearl necklace. We've got a red pearl and a pink pearl and a blue pearl and all kinds of pearls, okay? Actually, let's say we have 4 different color pearls because there's 4 different amino acids. Well, no, I take that back. We've got 20 different colors of pearls. There's lots of different pearls, okay? We have 20 different colors of pearls because there's 20 different amino acids. And so we'll bring in a pearl, okay? And that pearl represents 1 amino acid. And then we'll bring in another pearl, and that's another amino acid. And then we'll bring in another pearl, and that's another amino acid. And then we'll bring in another pearl. So the transfer RNA is the one that's bringing in the pearls. And then when we put all of these pearls together, that makes our protein, okay? Each of these is an amino acid. And we string them together to make our protein. All right? And we know what color to bring in because that's coded for on the mRNA. We're not just making a white strand of pearls. We've got a special color combination to match our outfit or whatever. Okay? Because the mRNA has a special amino acid combination to make a certain protein. All right? Just like you got to follow a cookbook instructions in order, and you got to have the right ingredients in the right order, you have to have your right amino acids in the right order to make the right protein. And that's really important. Okay? So, let's walk through these steps a little bit slower. Let's talk about transcription first. Okay? Remember the transcription part was the DNA to RNA. It happens in the nucleus. All right? So, a segment of DNA is copied into a complementary strand of mRNA. And what I mean by complementary is it's the base pair. Okay? So, if I have a set of DNA that says A, T, A, T, T, G, C, I'm going to make an mRNA strand that is complementary to that. So it's going to have the opposite base. If it's got an A, it's going to have a T on the mRNA. Except mRNA doesn't have T's. It's actually going to have a U. Okay? A, U, A, C, G. That's what the mRNA would make. Okay? Notice there's no T's. There's U's. Uracil is in RNA instead of thymine. There is no thymine in RNA. Okay? So, just like in DNA, we had that enzyme called DNA polymerase that brought in the matching letters. We have a very similar enzyme in transcription called RNA polymerase that does the same thing. It brings in the correct bases that lines up according to the sequence. Okay? Just like we had that DNA split open In the fork, we have the same thing in transcription. That was in replication, okay? But now we're talking about transcription. But we have the same thing that happens. The DNA opens up and we make a complementary sequence of RNA, okay? Now, in replication, we copied the whole cookbook. We copied the whole DNA molecule, all 6 feet of it, okay? In transcription, we are trying to make one protein. We are trying to make one recipe from that cookbook. So we're not going to copy all 6 feet. Maybe we'll copy 3 inches. We're just going to copy a short segment of that DNA. Specifically, the gene or the piece, the part that has the instructions for making whatever protein we're trying to make. Okay? And so RNA polymerase helps to do that. And then we make that version called mRNA. Again, this is happening in the nucleus because that's where the DNA lives and it never leaves. All right? So, when we put these letters together, these nitrogen bases, these A's and T's and G's and C's, we make something called a codon. So a codon is a group of 3 nitrogen bases. And 3 bases code for a specific amino acid to add to as a pearl, right? To add to our protein that's being grown. So, here, when we read these sequences, we would divide it up. We would read UCG, CAC, and GGU, 3 bases at a time. And then each of those 3 codons code for a different amino acid. UCG makes the amino acid serine. CAC makes the amino acid histidine. And GGU makes the amino acid glycine. And so those amino acids would be brought in by that enzyme we just talked about, RNA polymerase, and added to the growing protein chain. Okay? One pearl, another pearl, another pearl. So we've added 3 more amino acids onto our amino acid pearl necklace of a protein. Okay? Now, this is a diagram to tell which base makes what. Okay? So, for example, UCG. If we look at UCG, UCG is going to make serine. That's what I had written down here. CAC is going to make histidine. Okay? Each one of these on the outside is an amino acid. There are 20 amino acids. Okay? Some of them are coded for by more than one thing. So CUU makes leucine, CUC, CUA, CUG. All of those make leucine. Okay? Some of them may only be coded for by fewer numbers, fewer things. So AAC makes asparagine, AAU makes asparagine. If you change it to AAG or A, then you get lysine. And that's different from asparagine. Okay? So these are the 20 amino acids coded for by a codon. Okay? Codons are mRNA. This sequence you read from the mRNA, not from the dRNA. That's why it's got the U in it, because the U comes from the mRNA. All right? So, there's one codon, AUG. Let's look at it here. AUG codes for methionine. Methionine is the start codon. So methionine tells you to start making your amino acids. So you've got a methionine, and then you've got a code for a codon, and then you'll go until you'll see a stop. And that's the end. That's the end of that proton. This space in here is a gene. There's going to be several codons in there though. Okay? So you'll have a methionine, and then you'll have a bunch of letters that make a bunch of codons that make up a bunch of amino acids, and then you'll have a stop. And that segment between a start and a stop is a gene. And that makes one protein. Okay? Maybe 100 or 1,000 or a couple thousand bases long. Okay? Now, there's 3 stop codons. We see one here, UGA, and then we see the other tier here, UAA and UAG. Those are our stop codons. Okay? So between a start and a stop is where you have a gene. All right? Now, let's talk about translation. Translation is converting, remember, mRNA to a protein. But remember, the proteins are made up of amino acids. So really you're building the amino acids first. That is going to happen on the ribosome. Proteins are built on a ribosome. Okay? So, after transcription, we said transcription happened in the nucleus. So here it is. The mRNA leaves the nucleus through these holes called nuclear pores. It leaves the nucleus and it goes and binds on a ribosome. Okay? And then the ribosome is going to read these bases. It's going to read this first one, AUG, that's our start. So once it finds that start, it's going to know that it's going to start making the bases. Then it's going to read UUC. And it's going to build whatever amino acid UUC codes for. UUC is going to be phenylalanine. So it'll make a phenylalanine there. Then it's going to read AAA. AAA is going to make lysine. So it'll put a lysine there. And it's going to go and go and go until it hits a stop. And what it builds here, once all of those amino acids are linked together, is your protein. Okay? That's going to be your protein. This is happening on the ribosome. This is the mRNA. Notice that it's single-stranded, but it's the complement of 1/2 of the DNA, which it made in transcription in the nucleus. It copied down the instructions, okay, and brought them out here to be built. All right? So, let's talk about that other type of RNA. The rRNA, okay, that makes up our ribosome. So here's our ribosome, this blue and this orange part. There's 2 pieces of the ribosome, a big part and a small part. So we're not going to get into all that. But we've got our tRNA here that's really important. So what the tRNA does is it brings in the amino acids. Each tRNA, this one's not showing one, but it has an amino acid on the end. And it brings in the amino acid that is coded for by its bases here. Now, these bases on the tRNA are called anticodons. They are going to be opposite of the bases on the mRNA. So here's our mRNA. mRNA says CUG, so the tRNA is going to say, opposite or complementary to that, GAC. Okay? Now, it's still RNA, so it's still going to have uracil and no thymine. But this tRNA, right here, this is our tRNA, kind of looks like a T. It carries this amino acid that codes for this, the part on the mRNA. Not the part on the anticodon, the part on the mRNA. So CUG, whatever that is, CUG is leucine. This guy right here is leucine. Okay? So he's bringing in that pearl for the pearl necklace. And then we put all those pearls in our pearl necklace, and they form a polypeptide, which is also referred to as a protein. Okay? A polypeptide or a protein. All right. And this is going to go on and on. The tRNA is going to keep bringing in bases until it hits that stop codon. Okay? And at that point, that'll be cut loose and the protein will go free. All right? So, here we've got the whole process here. We've got our single strand of DNA right here. Remember, it has another side to it, right? But we separated it, and we're just using one side. So that single strand of DNA is made a complementary, meaning the base pairs are opposite, strand of mRNA, which has the codons on it in groups of 3. And these codons code for the amino acids, which are here, that are used to make a polypeptide or a protein. Okay? This part is transcription. This part is translation. In the nucleus and in the ribosome. Okay? So, just to reiterate here, the sequence of bases in DNA is used as a template for mRNA. The codons of the mRNA specify the amino acids. And then the genes, which is a segment of the DNA, contains the instruction for the proteins. And many proteins are enzymes, such as these polymerases that we've been talking about. And they are helpful in chemical reactions. Okay? You're dealing with Kai, and you're working with his situation, and he has a faulty protein. And so a lot of times in health and medicine, we have these faulty proteins because something has gone wrong in this process. We've had some type of mutation, and that's what we'll talk about more tomorrow. Okay? So I think that's all for today. No, that's a lot of information. Okay? I know that's a lot of information. I hope you paused along the way, wrote down a few notes. If you have any questions about it, please feel free to ask. Go back and answer those questions in the Google form. And then also, I would go ahead and go back and watch that 4-minute video as well, just as a refresher, just to kind of see it all together from another perspective. Okay? Let me know if you have any questions. If not, then you'll have a great day.